In this video, we are continuing our unit on organic molecules or biological molecules. We are moving on to proteins. So proteins are these little molecular machines, or like these little molecular robots that get everything done in the cell and in your body. They have multiple roles. Some examples of roles they take are defense. An example of a defense protein are antibodies. Also antimicrobial peptides. Your cells not only make antibodies as little chemical weapons against pathogens, but you make a variety of proteins that are your own internally made antibiotics. So proteins play a role in defense, in communication. Many hormones are proteins, neurotransmitters. Those are all kinds of proteins. Structure. So proteins are for structural support. The cytoskeleton, like the skeleton of the cell, is built out of protein. Movement. So your muscles move because of muscle fibers, which are made out of protein. Flagella on bacteria and on sperm, they're made out of protein. So proteins can be used in movement, for portals across the cell membrane. So the cell membrane is a barrier between the inside and outside, but there's things that need to move across, like waste needs to move out and nutrients need to move in. Those are going to move through protein channels. And also proteins can be enzymes, which build, break down, and change molecules inside the cell. So to build these polymers, we're using enzymes. To break down polymers, we're using enzymes. To conduct any chemical reaction, to change any molecule from one type into another, we're using enzymes, which are also a type of protein. It's a common misconception that we eat proteins for energy, because we think this because we get these protein bars, and protein bars are supposed to be like good energy for going to the gym. Protein bars have energy in them because they're filled with sugar, sometimes fats, but mostly sugar. The protein in protein bars is for building muscle, for the movement component. Proteins are important in fitness and health because they're the dynamic parts of your body. They're the robots that get everything done. Now, proteins are encoded by your DNA. And we've talked about this before. DNA does not code for lipids. It does not code for carbohydrates. It codes for proteins. And then proteins, like enzymes, build the other molecules of the cell. And the proteins even build the DNA. So it's a circular, like DNA codes for protein builds DNA. DNA codes for protein builds DNA. So these are the dynamic components of the cell. Now the shape of a protein, its form, its three-dimensional shape, determines its function. There is this diverse set of three-dimensional shapes and complex folding patterns in proteins. The monomers, so proteins are polymers. The monomers are amino acids. So a protein, which we call a polypeptide, is a string of amino acids. There are 20 different kinds of amino acid, and they're arranged in these different sequences to result in all the protein forms and functions that we have in living creatures. So here's an example of a three-dimensional shape of a protein. So this is a string of amino acids, and the amino acid chain will loop around back on itself to make these complex three-dimensional shapes. So this is a ribbon model of a protein called lysozyme. Now the shape of an enzyme matters because it determines its function. So all enzymes have grooves. We also call them active sites, and they are the perfect shape and the perfect chemistry to bond to a very specific molecule. Once bound to their substrate, the enzyme is going to cause a chemical reaction to happen. So this particular enzyme called lysozyme, this is an enzyme that you find in your tears and other body fluids, and it is the perfect shape to bind to a very specific molecule on a bacterium's surface and destroys that molecule, and that kills the bacteria. This is just a different model of the same enzyme, of the same molecule. This is a space-filling model instead of a ribbon model. All right, so I told you that proteins are polymers. They are amino acids strung together. This is what the monomers look like, the individual amino acids. So amino acids always have the same basic structure. There's the amino group, That's one of the functional groups we learned about. And then a carboxyl group, that's another one of the functional groups we talked about. And so the amino group of one amino acid undergoes a dehydration reaction with a carboxyl group of another amino acid, and they string together back to, amino group to carboxyl group, amino group to carboxyl group, and so on and so forth. Now, I said there are 20 different amino acids, and what makes each of those 20 amino acids unique is what's here in the R group. R is not an abbreviation for an element in the periodic table. It's a placeholder, kind of like the letter X in algebra. So this R group can be any number of things. There's 20 different R groups, and that's what distinguishes one amino acid from the next. Different R groups have different chemistries, and so they give the different amino acids different properties. So here's a diagram of the dehydration reaction. And one thing I want to point out is, all the dehydration reactions we've looked at up to this point, it's a hydroxyl group that undergoes a dehydration reaction with another hydroxyl group on the next molecule. Here, there's only one hydroxyl group. It's okay though, because we have a hydrogen here, because remember what we're forming is H2O. So all you need is one oxygen and two hydrogens, and we have that here. We have the oxygen and one of the hydrogens in this hydroxyl group that's part of the carboxyl group, and then a hydrogen here. So this hydrogen links up with this OH to make H2O. This carbon now bonds with this nitrogen, right? And that's what we call a peptide bond, So instead of calling proteins polyamino acids, that's just like long and wordy, we call them polypeptides. That is a nice, alliterative sound to it. Polypeptide is another word for protein. In these polypeptides, in these proteins, you're going to have an N terminus on one end and a C terminus on the other. So the N terminus, because you're linking, carboxyl to amino group, carboxyl to amino group, carboxyl to amino group over and over and over again. But the ends, they still have their free amino group and free carboxyl group. So where the free amino group is, that's the N terminus. Where the free carboxyl group is, that's the C terminus. Now, the R groups are what give each amino acid its unique properties. Some R groups are nonpolar. So you see there's a whole bunch of hydrocarbon R groups. Those are nonpolar. Also, these sulfhydryl groups are nonpolar covalent bonds. So these are nonpolar. So here's your nonpolar amino acids. Now, remember, nonpolar molecules, do they dissolve in water or don't they dissolve in water? Are they hydrophilic or hydrophobic? Remember, for something to dissolve in water, it has to have some sort of positive or negative electromagnetic charge, or both. And then we have our ionic amino acids. Some are positively charged, some are negatively charged. And we have our polar amino acids that are built, where the R groups are built of polar covalent bonds. So polar has positive and negative charges. Ionic has positive and negative charges. Those are going to be the hydrophilic R groups. Now, if you don't know if an R group of an amino acid you're interested in is hydrophilic or hydrophobic, remember all you need to do is look at the elements that make up the R group, the carbons, hydrogens, sulfurs, whatever's there, and compare their electronegativities using an electronegativity chart. Remember that if two elements are bound to each other that have an equal electronegativity and equal pull of electrons, it will be nonpolar, and there will be no development of negatively charged regions and positively charged regions. On the other hand, if you have elements that are sharing electrons that have really unequal electronegativities, where one element is really strong and one element is really weak in its attraction for electrons, that's where you get these collections of negative charges in one part of the molecule and positive charges in a different part of the molecule. So to figure out if an amino acid is going to dissolve in water, you have to figure out if it's hydrophilic. It will be hydrophilic if it's ionic, if it has positive or negative charges, or if it's polar. To figure out if it's polar, you look at an electronegativity chart to figure out if you have inequalities in electron sharing. So these amino acids link up together, amino group to carboxyl group, amino group to carboxyl group, and the R groups stick out the sides with their unique properties. The R groups of non-adjacent amino acids in the chain can interact with each other, forming ionic bonds with each other, forming hydrogen bonds with each other, and that's going to cause the polypeptide to fold up into a three-dimensional shape. So it's interactions between R groups really that hold the three-dimensional shape of a protein together. Now, there are 4 levels of protein structure. There's primary structure, secondary structure, tertiary structure, and quaternary structure. So let me go through those one by one. Primary structure is just the sequence of amino acids in the polypeptide chain. So, like it could be serine, leucine, aspartic acid, serine, tyrosine, et cetera, et cetera. It's just the sequence. It's the order of the amino acids. Now, because these R groups are sticking out the side, each R group has its unique chemical properties. The sequence of amino acids, the primary structure, it determines what the chemistry is in different parts of this amino acid string. There's like an there's an ionic region, there's a hydrophobic region. So it's the sequence of amino acids, the sequence of R groups that dictates the chemistry that you find in different parts of this polypeptide chain. All right, secondary structure. So hydrogen bonds form between the amino group and the carboxyl group of non-adjacent amino acids. Remember that covalent bonds form between the amino group and carboxyl group of adjacent amino acids, between neighbors, right? And that's through dehydration reactions. But remember that these are polar covalent bonds. So nitrogen hogs electrons from hydrogen. Oxygen hogs electrons from carbon and from hydrogen. So hydrogen is partially positively charged in both the amino group and the carboxyl group. Oxygen always has a partial negative charge. So there are these weak attractive forces between the hydrogens of the amino group and the oxygens of the carboxyl groups. As the polypeptide chain gets long enough to fold back on itself and form loops, these hydrogen bonds between non-adjacent amino acids are going to form. And 2 shapes are held together by these hydrogen bonds. There are beta-pleated sheets, which you see down here. This is a beta-pleated sheet where it's just, you have this polypeptide chain and it loops back and they lay on top of each other. And they are held together by, again, there's a hydrogen bond formed between the carboxyl group of this amino acid up here and the amino group of this amino acid down here. So that hydrogen is partially positively charged and that oxygen is partially negatively charged. So they have these attractive forces between them. And also the second shape that's formed by these hydrogen bonds are alpha helices. So a helix is a spiral shape. And that is also held together by these hydrogen bonds between the partial positive on the hydrogen from the amino group and the partial negative on the oxygen from the carboxyl group. All right, so back to those R group side chains that are sticking out. So those R groups can interact with each other. So different amino acids can interact with each other through their R groups in the chain as it folds up upon itself. And that's going to determine a lot of the 3-dimensional folding pattern of a particular protein. So for example, ionic bonds can form between positively charged ionic R groups and negatively charged ionic R groups. So cations and anions. So here you see that here. You have an ionic bond. So this R group is ionic with a negative charge, and this R group is ionic with a positive charge. And so those attractive forces, they're not sharing electrons, but they're just attracted to each other like sodium chloride in salt. You can also have different amino acid R groups that form covalent bonds with each other. This usually happens between sulfhydryl groups, so between sulfurs of different amino acids. So it's called a disulfide bridge. 2 sulfurs, one from each of the different amino acids, forming covalent bonds with each other. And then you can have hydrogen bonds form between polar R groups. There were plenty of polar R groups. And so those will tend to form hydrogen bonds with each other. And then you can have these hydrophobic interactions. So remember that these proteins are floating around in water because most of the cell is water. And so the hydrophilic R groups will be attracted to water. They'll tend to be more superficially located on the polypeptide chain in this, ball of this knotted ball of protein. And the hydrophobic R groups will tend to cluster together. They'll be excluded from water. They'll be excluded from those attractive interactions. So they're all going to cluster together in certain parts. So put all of these level of structures together and you get these really complicated three-dimensional shapes. You have these alpha helices in some parts. You have the beta-pleated sheets in other parts. And then superimposed on top of that, you have the R group interactions of tertiary structure. And that creates these really complicated three-dimensional protein shapes. So here's just another image of the same. Here you can see water is attracting some of the hydrophilic R groups so that they're more superficial. You see those hydrophobic interactions clustered more towards the inside of the protein. So the primary structure, the sequence of amino acids, determines the tertiary structure because the primary structure determines where you find different chemical properties in the chain, and that determines how the protein folds. So remember that DNA is the code, the instructions, the blueprint for building proteins. So if an amino acid in the primary structure is changed to a different amino acid, or deleted due to some mutation in the DNA code, this can sometimes result in a dramatic change to the three-dimensional shape of the final protein. And remember, shape equals function. So a change in the shape of a protein is going to change the functionality of that protein. So for example, you have these ionic bonds here. But what if you had a mutation in the DNA code and this positively charged amino acid was changed to a hydrophobic amino acid or even a negatively charged amino acid? If this positively charged amino acid became a negatively charged amino acid, instead of these two attracting each other, holding these two loops close together, now they're going to repel each other. That's going to completely change the shape of this protein. So that explains how changes in the DNA code can change a living organism, because remember, these proteins are the little robots that get everything done in the cell. And if there's one protein that can't do its job or that's doing its job differently than it used to, that might have dramatic consequences for the living organism. Quaternary structure describes how more than one polypeptide chain can interact through their R groups and be held together through their R groups. Many proteins include more than one polypeptide chain. So here's one example. This is a G protein. G proteins are receptor proteins on the surfaces of your cells. They receive chemical signals, like hormones, that come from outside the cell, and they transfer the message to the inside of the cell, causing the cell to begin behaving differently in response to the chemical signal. Through the interaction between hormones and receptor proteins like G proteins, a hormone that's released by one organ in your body, like your brain or your adrenal glands, those hormones can alter activities of other organs, of other cells in your body. These G proteins are only one of many examples of proteins in your body that are built of more than one polypeptide. Now, primary structure also dictates quaternary structure because, again, it's R group interactions that hold these chains together. So a change in amino acid sequence can result in a change in shape or even a complete inability for the different polypeptides to join together properly. So remember, again, shape equals function. Different G proteins are designed to receive different chemical signals, and the active site has to match the shape of whatever hormone it's designed to receive perfectly. So a change in the shape of a G protein, for example, could mean that the G protein is no longer able to receive a particular hormone signal. So hemoglobin is another famous example of all four levels of protein structure. Your red blood cells, they are just sacs of hemoglobin. So iron is carried inside each of these four subunits, and the iron binds to oxygen to carry it through your body to all the cells in your body. Also, hemoglobin carries carbon dioxide from your cells. Carbon dioxide is a waste product made by your cells. So it carries carbon dioxide from your cells back to your lungs for exhalation. So here you can see all four levels of protein structure. So primary structure is just the sequence of amino acids in the chain, straight line. Secondary structure is held together not by R group interactions, but by hydrogen bonding between the amino group of one amino acid and the carboxyl group of another amino acid that is not its neighbor in the chain. And you get these alpha helices or beta pleated sheets. Here's tertiary structure. Tertiary structure is held together by the R group interactions between different amino acids in the polypeptide. And then quaternary structure is also held together by R group interactions, but it's between different polypeptide chains.